Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So we started discussing about immunology, where we discussed about the different types of immune systems. Then we started discussing about the cells of immune system, right? So we discussed about the innate immunity. We discussed about adaptive immunity. Innate immunity is the type of immunity which we possess since birth, whereas the adaptive immunity, which is specific and it develops over the time due to the exposure to different experiences, due to the exposure to different pathogens. So we learned about hematopoiesis, the process of generation of blood, the mature blood cells from the hematopoietic stem cells. So we learned about the characteristics of hematopoietic stem cells and how it gives rise to the different lineages that will give rise to the different cell types, which are the cells of the blood and the cells of the immune system. So we discussed about how those cells, this hematopoietic stem cells is giving rise to lymphoid lineage and the myeloid lineage, where the cells of myeloid lineages as well as the natural killer cells that belong to the innate immunity and the cells of lymphoid lineage belong to the adaptive immunity. So then we discussed about how those hematopoietic stem cells they get generated and matured from the bone marrow and as well as in the peripheral tissue. Then we started discussing about organs of the immune system. The organs they are going to provide the microenvironment for those immune cells to originate from the from the hematopoietic stem cell, as well as they are going to provide the microenvironment for the maturation of those immune cells. There are certain organs which does not help in maturation or generation of those immune cells, but rather than this job they provide a microenvironment for those immune cells to interact with the antigen and to initiate the immune responses. Such lymphoid organs are known as secondary lymphoid organs. Okay, so lymphoid organs are the organs of immune system, and based on what job they do, they are categorized into two different categories. The primary lymphoid organs that we have discussed in previous classes. So the primary lymphoid organs are the ones where those immune cells they are originated and they get matured. So generation and the maturation of immune cells will be seen in the primary lymphoid organs. So we talked about the primary lymphoid organs such as bone marrow, which will give rise to the B cells, the dendritic cells, macrophages, means the cells of myeloid lineage. And we talked about thymus, where the T cell precursors from the bone marrow they are going to go to the thymus and they will get matured into the naive T cells. Okay, so those two are the primary lymphoid organs, and today we are going to discuss about the secondary lymphoid organ, lymph nodes. Okay, for that matter of purpose, we will first know about what is lymph, what is the lymphatic system. We'll talk about lymph nodes, the specialized structures which are also referred as secondary lymphoid organs. We have discussed about spleen, which is also a secondary lymphoid organ, and today we will be talking about lymph nodes. And there's one more secondary lymphoid organ or the secondary lymphoid tissue, which is known as MALT, mucosal associated lymphoid tissue, which is less organized compared to the spleen or the lymph nodes. So the secondary lymphoid organs, there are multiple. Okay, first is spleen. We have discussed about spleen in previous class. Then there's malt, okay, that we will be discussing in next class, and in today's lecture we will be discussing about lymph nodes. Okay, so let's get started with the lecture. So the all of the lymphoid organs that I discussed now, they share some common features. Okay, so just a second. So the secondary lymphoid organs are spleen, malt, and lymph nodes. Okay. I'm writing mucosal associated lymphoid tissue separately because it is less organized compared to the spleen and nodes. So all of those three, they are called secondary, they are referred secondary lymphoid organs because they share some specific features. And those features are that they obviously all of them they are important for initiation of immune responses, but other than that, all of them, they have separate zones for B cells and T cells. Okay, so these are the features that belong to the secondary lymphoid organs, is that they have separate zones for B cells and T cells. Okay, because B cells, when the antigen will be presented to the B cells they will get mature and they are going to form plasma cells, which are the antibody secreting cells. And if the T cells they are interacting with antigens, they will be they will mature and either they will form CD8 positive T cell, the cytotoxic T cell, or they are going to form the CD4 positive T cell, the T helper cells, and now they are going to mediate the pathogen killing. So that is why the separate zones exist in the secondary lymphoid organ for B cells and T cells. So the niche for B cells and T cells that will be separate in those tissues, in those secondary lymphoid organs. And this is common in all of those organs. We talked about spleen and we discussed about how how in the spleen there will be follicles where those there will be follicles where those cells, those B cells will be situated, and then there will be marginal zone, and there will be the periarterial lymphoid sheath. So this three, they belong to the white pulp, and here the immune responses will be initiated. So the periarterial lymphoid sheath is the one which is T cell zone, whereas the follicles, the primary follicles, is the B cell zone. In the same way, in the lymph nodes, there will be the layers, different layers, which will separate the B cell niche from the T cell niche. Okay, so this is the common feature that belongs to all of the secondary lymphoid organs. Other than that, those secondary lymphoid organs, they are tiny. They will have tiny follicles. So the areas where the B cells are going to grow and they are going to produce the antibodies. So the B cells will get matured to form the plasma cells, which are going to produce high affinity antibodies. Okay, so this is also the feature that is shared by all of those lymphoid organs, that they are going to have lymphoid follicles. Okay. So as we discussed in last class, spleen has the primary follicles, and those primary follicles, when the B cells mature in them and they form germinal centers, they are referred as secondary follicles. In the same way, lymph nodes also have primary follicles. Even the malt is going to have the lymphoid follicles. Okay, so the lymphoid follicles, these are the tiny regions where the B cells are going to interact with the antigens, and they are going to get matured. So when the B cell, so this primary those lymphoid follicles are B cell rich. So when they are B cell rich, they are referred as primary follicles. And once they interact with the antigen and they get activated and they get matured to form plasma cells, which are going to secrete the antibody, they are referred as the secondary follicles. Okay, so secondary follicles are the ones where the germinal centers will be present. Germinal centers are basically the areas where the B cells are going to get activated and they are going to produce the antibodies. Okay, so this is the basic difference between primary follicles and secondary follicles. Primary follicles do have B cells, but the secondary follicles, they have activated B cells, the ones which are going to produce the antibodies. So this is the common features that will be shared by the secondary lymphoid organs. But there is difference, right? And the difference is, first of all, those are different organs and they are situated at different locations. But other than that, there are a few differences between all those secondary lymphoid organs, and that is about organization. Okay, so the spleen and lymph nodes, they are highly organized tissue where they are going to have fibrous capsule which is going to encapsulate those organs. So both lymph nodes and spleen, both of those organs, they are going to have fibrous capsule. We have already discussed about spleen. Okay, spleen being a soft and fist sized organ which is encapsulated by fibrous tissue, a fibrous capsule, whereas lymph nodes are also small bean-sized nodes which are encapsulated by the fibrous capsule. But malt, mucosal-associated lymphoid tissue, it is less organized and it is present on the areas where the where the internal organs, they have the chance, they have the point of contact to the external environmental factors. Okay, so we'll discuss this about in the lecture, for a separate lecture for mucosal associated lymphoid tissue. We'll discuss about Peyer's patches, we'll discuss about tonsils. So all of them, they are less organized compared to them, and they do not, the malt tissue, they do not have this fibrous capsule. So this is the difference between all of the secondary lymphoid organ, but basically all of them has one job, and that is to initiate the immune responses. Other than that, spleen also has some other roles, that is to filter the blood, to remove the old and damaged RBC, whereas lymph node is fully dedicated for the for the immune responses, for initiation of immune responses, right? So when we discuss about immune cells, we discuss that those, all of them, they develop from the hematopoietic stem cells, and they develop from hematopoietic stem cells in the bone marrow, right? And the maturation of those cells from the precursor cells will be done in the peripheral tissue. And once those immune cells mature, they are present in our blood, and they circulate in all over body. So by now, from the high school standard, from high school level, you might already know that we have two circulatory systems in our body, so that the nutrients, so that these cells which are needed at the tissue, they will be circulated in all over body. Those two circulatory systems are the blood system, the vascular system, and the lymphatic system. Okay, so the blood system, we have talked about that in very detail. All of you know about that. And there is lymphatic system. So I'm going to talk about lymphatic system now in detail, because it is very important, and it is kind of a highway for the immune cells to reach to the tissues, and it is also a path for all of those antigens which are there, which have invaded our tissue, to reach to the immune cells. Okay, so that is why we are going to discuss both of those. So blood system, the as well as the lymphatic system, both of them, they provide a kind of highway for those antigens as well as immune cells to travel across the body. So immune cells are going to reach to all of the tissues in the body using those pathways, and the antigens which have been invaded through in the tissue, they are going to reach to those immune cells using those pathways. Okay, so it's like, imagine immune cells as the patrolling cops, or simply the cops who are going to who are whose job is to see whether the infection is there or not. So the infection, I told you that the infection, or the pathogens, they could enter into your body through wound or through injury. Means they can enter through the blood, or they can enter through the open cavities, the oral cavities. They can enter through the urogenital tract. So those, or the respiratory tract. So they are going to enter through those open areas, or they are going to enter through the blood. So antigen could be present over the in the blood, which are referred as blood-borne antigen, which will be taken care by the spleen. Okay, the spleen will be trapping those antigens, and it will mediate the killing. Okay, and how the cells kill those pathogens, we have talked about those mechanisms in particular cell lectures, right? So you already know that. Then there will be some antigens which are going to enter through those cavities, such as, let's say, respiratory tract, or let's say from the gastrointestinal tract. So there they are going to face the MALT, the mucosal associated lymphoid tissue, such as tonsils, such as Peyer's patches in the gut. They will be facing, they will be facing those tissue, mucosal associated lymphoid tissue, which will have immune cells, even in the appendix. Okay, so tonsils, Peyer's patches, appendix, or the lymphoid patches in the bronchi. So all of them, they are providing a way to keep a check on the pathogenic attack in those open cavities. Okay, then let's say there's some injury, there's a wound. So from this injury, the pathogen can directly enter those tissue. Okay, and when this happens, or let's say through the blood circulation, the pathogen is entering in some tissue. Okay, due to the capillaries, because capillaries, they are going to leak through the plasma, and that in that plasma there could be those blood-borne antigens which can now invade the tissue. So in any cases, when the tissue is invaded by the pathogens, so now those, the tissue resident cells are going to take care of those pathogens. But besides that, those tissue resident cells, tissue resident immune cells, they are going to alert the other T cells, other B cells to, to get activated and to kill those pathogens. And that will be done in the lymph nodes. Okay, so lymph nodes are the sites where the tissue resident antigens, they will first interact. So this will be the first interaction, their first interaction with the lymphocytes. Okay, so whenever there is any pathogen in the tissue, they will be either tackled by the, antigen presenting cells which are resident in the tissue, or the macrophages which are already present in the tissue. So these macrophages or dendritic cells, they either will engulf those, those pathogens, or they are going to do the antigen presentation. So this antigen presenting cells, they will, take those antigens, or simply the whole bacteria, the particular those bacteria, those particles, they are simply going to travel to the lymph nodes where they are going to face the lymphocytes for the first time. Okay, so I hope the whole scenario is clear to you all. So lymphatic system is the system which collects a fluid, okay, from the surrounding tissue, which is known as lymph. Okay, and that is going to help into initiations of the immune responses. So let's first talk about lymphatic system, then we will discuss about lymph, and then we'll discuss about the lymph nodes. Okay, so yeah, I'm going to talk about lymphatic system. So there are a lot of differences between blood system and the lymphatic system. So you already know about blood system, right? So blood system is a fast network compared to the lymphatic system because it has a pump, which is heart, right? So the heart is pumping the blood, and so the arteries are going to pump the blood. So heart is going to pump blood through the arteries, and then that blood is going to go to the tissues, and tissues will be, using the nutrients present in the blood, and this deoxygenated blood will travel through the veins to the heart so that the blood is pumped again. So that is why this system is quite fast compared to the lymphatic system, because in the lymphatic system there's no pump present. Okay, so that pump, so only heart is only pumping blood, right? So blood system has pump, not the lymphatic system. So how does lymph travel through the body? So lymph travels through the body using the contractions of the smooth muscles of those lymph nodes, as well as the contractions of the skeletal muscles which are present as in the surrounding areas. Okay, so lymphatic system is a slower system, and again it is a one-way system. So only tissue resident, so interstitial fluid, when it enters into the lymph vessels, sorry, that is called now lymph, and that lymph will be drained out of the, drained into the lymph nodes from the tissues, and now that lymph will be again drained into the blood at the in the veins, in the left subclavian vein as well as in the right subclavian vein. Okay, so lymphatic system is the system which collects the interstitial fluid. Okay, so what happens is that, let's first talk about this. What is an interstitial fluid? So the blood is being pumped by the heart. So artery will form arteriole. Okay, the smaller the smaller version, and then they are going to form capillaries, which are, one cell thick. So capillaries are one cell thick. So RBC cannot escape through the capillaries, but the other, other substances such as the plasma and the nutrients, they can seep out of the capillaries. And when this, the capillary network, this capillary network is formed inside the, inside the tissue. So this is the network of capillaries. So capillaries network is formed inside the tissue so that the nutrient exchange is mediated through them. So during that, the plasma oozes out through through those capillaries, and almost 20 liters of plasma oozes out through those capillaries every day. Okay, and it is taken back. Okay, so it oozes out through those capillaries, and it again is taken back in those capillaries, and that is again circulated through the veins. Okay, so this is what happens in the tissues, but not all of this plasma will be taken, it will be taken in by the capillary. The 3 liters of the fluid stays behind in the tissue, which is known as interstitial fluid. Okay, so this fluid which stays in the tissue, it has little bit different composition compared to the blood plasma. So blood plasma, it compared to blood plasma, this interstitial fluid is, rich in multiple proteins. So this interstitial fluid, rather than so it is not being taken up by the blood system. So this interstitial fluid will be taken up by the nearby surrounding lymph vessels. Okay, so there will be lymph vessels which are one-way lymph vessels. Okay, so this lymph vessels will take up this interstitial fluid, and now this interstitial fluid, which is traveling, which is taken in by those lymph vessels, will be known as lymph. Okay, so lymph is the Latin word for clear fluid. So this is basically the blood plasma, but it has different composition from the blood plasma. So this lymph is going to have the different cells, the cells of immune system. Okay, it is going to have the blood plasma component, but it is basically rich in proteins. So it is going to have cells of immune system, and it is going to have other components. So this interstitial fluid, when it enters the lymph vessels, it is referred as lymph. So lymphatic system basically collects this lymph. Okay, so lymphatic system is the system that collects this interstitial fluid, means that collects interstitial fluid, which will be referred as lymph when it is taken in. So this lymph, it will enter the lymph vessels, and this lymph, collected lymph, will be drained. It will be drained into blood system. Okay, so so that the all of the interstitial fluid that was that stayed behind in the tissue, that will be taken up by the lymphatic system, and that will be again returned to the blood. So whatever blood lost, 3 liter of plasma, that blood lost, that will be returned to blood again. Okay, returned to the blood system again in the veins. Okay, so there is, this lymphatic system is made up of those lymph vessels, which will form lymph duct in future. Okay, so so artery is the thicker version, and it is going to carry the blood. So the blood pressure will be high, whereas capillary is thin, and it is one cell thick. Okay, in the same way, the vessels are thinner, and those multiple vessels, they are going to come together, they are going to form trunk, and then those trunks, they are going to come together to form ducts. Okay, so the lymphatic ducts are the ones which are going to carry those lymph, and those lymphatic ducts are going to drain this lymph into the blood system. So the largest lymphatic duct is known as thoracic duct. Okay, and this thoracic duct will be draining this lymph into the into the left subclavian vein. So thoracic duct is present on the left side of the body. So the thoracic duct is going to drain this lymph into the left subclavian vein, because here the pressure is very low. That is why the lymph can easily be taken in by the blood. Okay, in the blood vessels. And then the right, the right lymphatic duct, there's all of the majority of the lymph will be given into the blood system by thoracic duct, but there's right system as well. So the right lymphatic duct is going to drain the all of the lymph which is present in that duct into the right subclavian vein. So right lymphatic duct is going to drain the lymph into right subclavian vein. Okay, so this happens, and this is how the lymph which is collected and it is returned again to the to the blood system. So this drainage system, it keeps the fluids levels balanced in our body. So if this blood, if this interstitial fluid in the tissue, it is not collected by the lymph vessel, it's it accumulates in this tissue, and when the interstitial fluid accumulates in the tissue, it actually results into swelling, which can be which can be really dangerous for the person, for the individual. So this swelling due to the accumulation of interstitial fluid in the tissue is known as edema. okay, which is basically the tissue swelling due to the accumulation of interstitial fluid, and that is why lymph system is very, very important. So how does this, lymph flows through the veins, flows through the vessels? So when the lymph is taken in, the lymph, all of those lymph vessels, lymph ducts, they are one way. Okay, so means they are going to take take the lymph from the tissue and they are going to give it to the blood. This doesn't go other way around. Okay, the way, the blood is pumped from the heart and then it is brought back to the heart. Okay, so it can, it can travel from heart to the tissue, and it can also travel from the tissue to the heart. But unlike this blood system, the lymphatic system drains the, lymph in one direction, and that is why, just like the veins have walls, the the veins, okay, which are going to carry the blood, they also have walls. In the same way, the lymphatic vessels, they are also going to have one way walls. So the one way walls, they ensure that blood doesn't flow backwards, because basically the pressure is quite low. So arteries have all of the blood pressure, and veins do not have that much of the blood pressure, right? So that is why in the veins the walls are present. In the same way, lymphatic system does not have pressure, does not have a pump to, to pump the lymph, and that is why they are going to have one way walls, which opens only in one direction. So this is the wall and this is the valve. Okay, so this is the valve. So it opens only in one direction, so that the lymph flows in one direction. So it ensures the one way flow of the lymph. So those, those the lymph vessel, the ducts, they do have the smooth muscles. So due to the arterial pressure, due to the pressure that is created by the heart, or the, due to the smooth muscle contraction in those vessels, the lymph will be, carried forward through that particular vessel in one direction, and walls maintain that one direction. So smooth muscle contraction is ensuring the smooth muscles which are present in those, in those vessels, that ensures that the lymph flows in the given direction. And again, there are present, surrounding those lymph vessels, there are present skeletal muscles. So the contraction of those skeletal muscles will also, help in the movement of the lymph. So that is why we, the exercises, walking, or even breathing, whenever any exercise which involves muscle contraction, that is going to help into the lymphatic flow. So lymph flow is, passive, and it will be controlled by the muscle contraction, the skeletal muscle contraction, and it is, that will help the lymph to flow better. That will help physically lymph to flow better. So this is how the lymphatic system which lacks any pump will also have a nice regulation due to the due to the skeletal muscle contractions. So we have talked about lymphatic system in detail. We also talked about lymph. So now let's discuss about how the antigens and immune cells, they are going to travel through this lymphatic system. Okay, so for that matter of purpose, let's first learn about the lymph nodes. Okay, so let me draw a schematic for the lymph nodes. So lymph nodes, they are small lymph nodes. They are small bean-shaped structures which are present alongside the lymphatic system. Okay, so these are the small bean-shaped encapsulated structures. Okay, so let's draw a schematic. So this is the lymph node. Okay, so in this lymph node, there will be entry of the lymphatic vessels directly. Okay, so these are the afferent lymphatic vessels. Okay, means these are the afferent lymphatics, means they are going to take the lymph inside the lymph node. So this means that lymph is incoming through those lymph is coming inside the lymph node through those vessels. So these are the afferent lymphatics, and this is the efferent lymphatics. Means this is going to take away the lymph. Okay, and then there will be blood vessels which are going to enter into those lymph nodes. So there will be blood vessels, which is this is the arteriole. Okay, so this is going to take in the blood. Okay, so this is bringing in the blood, and there will be system to bring out the blood. Okay, so these blood vessels are going to bring in the lymphocytes. They are going to bring in the antigen presenting cells. Okay, and even the blood-borne antigens inside this lymph node. So this lymph node, this has layers just like onion. Okay, so there will be outer layer, there will be a middle layer, and there will be the innermost layer. So just like onion, it has multiple layers which are present in concentric forms. So the outermost layer is obviously known as cortex, and in the outermost layer, in the cortex, there will be present follicles, okay, which are known as primary follicles. I have already discussed about this previously. So the primary follicles, they are present in the cortex region. So this region, this outermost region is cortex, and then there's middle region, which is known as paracortex. So this middle region that I'm representing by blue color, okay, so this particular region, okay, this is the paracortex, and just the innermost layer is known as medulla. Okay, let me draw the medulla in different color. So this innermost layer is medulla, and those blood vessels that are entering into those lymph nodes, they are penetrating capsule. So this is the outermost is the capsule, and they are also releasing their lymphocytes in the medullary region. So these are the three regions. Okay, cortex, the outermost one, then there is paracortex and medulla, innermost one. Okay, so the naive lymphocytes, they are going to come through the blood vessels inside the lymph nodes, and they will interact with the antigens here. And once they have interacted with antigens, they will be taken out from the blood vessels again. Okay, whereas the afferent lymphatics, they are going to bring the antigens as well as the antigen presenting cells. So antigen has, the infection has been there in the tissue. So let's say that it is a bacterial infection. So the part, either the afferent lymphatics can either bring the whole bacteria, the bacterial particles, okay, or it can bring the processed antigens through the antigen presenting cells. So either antigen or antigen presenting cells, they will be coming inside the lymph node through the afferent lymphatics. And once they interact with the antigens here, once the inside the lymph nodes, either B cells are going to interact with the antigens or T cells are going to interact with the antigens. So this activated T cells or activated B cells, they will be taken out from the lymph nodes through the efferent lymphatics. Okay, so that those activated cells, they can again go through the blood system, through the lymph to the blood, and through the blood to the tissue, so that they can kill those pathogens inside the tissue, or even if there's blood-borne antigen, they can kill that inside the blood. So those activated B cells and T cells, they will be taken out from the efferent lymphatics. But not just B cells are going to taken out from the efferent lymphatics, but the B cells which are activated, they will also secrete antibodies. So either the activated B cells will flow through this efferent lymphatics, or the antibodies, they will be secreted inside the blood vessels. Okay, so inside the blood, those antibodies will be secreted and they will be taken out. So either way, it is this is ensuring that whatever system is being, whatever immune cells they are getting activated, they will reach to the tissue. So how does antigen travel? So antigen, it will be, it will travel through the afferent lymphatics. So antigen as well as antigen presenting cells, they are going to travel through the afferent lymphatics inside the lymph node. Once they travel inside the lymph node, they will be trapped by the lymphatic system and they will be identified. So they, if the blood vessels are bringing the antigens, So the blood vessels are they are penetrating capsule as well. So the blood-borne antigens they will be trapped inside this capsule. Or if lymphatics are bringing the antigens, they are going to get trapped inside the cortex. Okay, so here those antigens will be trapped. All the antigen presenting cells they will be interacting with the cells which are with the B cells or T cells which are present inside the lymph nodes. So as I already discussed that the B cell zone and T cell zone is different inside inside the secondary lymphoid organs. So the follicles, so the cortical region is the one which is going to have B cells, and alongside that it will have follicular dendritic cells, right? So you remember when we discussed about dendritic cells, we discussed about follicular dendritic cells, we discussed about plasmacytoid dendritic cells, right? So one of the type is follicular dendritic cells, which is going to help into B cell development. So these are the specialized antigen presenting cells which are going to play a role in the B cell development. So those cells will be found in the cortex region. So in the cortex region, we can also call it the B cell rich region. Here the B cells are going to here the naive B cells will be present in the primary follicles. They are going to interact with the antigens and they are going to get activated, and they are going to form plasma cells to secrete the antibodies inside the blood, okay, in the blood system. So the cortical region will have B cells which are naive B cells, okay, which will interact in the follicles with the antigen. And besides the B cell, it will have follicular dendritic cells which are again helpful in the activation of those B cells. So follicular dendritic cells are going to do the antigen presentation to the B cells so that the B cells get activated here. So the cortex is the B cell rich region, whereas the paracortex it is rich in T cells. Okay, so paracortex is going to have the naive T cells in it, and here there will be naive B cells. Okay, so paracortex is the T cell rich region, and the medullary region is the one where the other cells such as macrophages or the plasma cells which are secreting antibodies will be present. So in the medullary region, it is not really enriched in any type of cell, but this is the region where the plasma cells are going to secrete antibodies, which will be taken in by which will be taken in by the blood system, or whatever lymphocytes are activated, they are going to exit through the medulla. So we can call that medulla is the exit zone. Okay, medulla is the exit zone for activated lymphocytes. And let's say if there's no antigen, then those lymphocytes will not get activated. So either activated or naive lymphocytes. So whatever lymphocytes are present in the lymph nodes, they will be taken out from the medulla through the efferent lymphatic vesicles. Okay, so medulla is also known as exit zone. So it also has lymphocytes, but lymphocytes are fewer compared to the cortex or the paracortex. So cortex and paracortex are the lymphocyte rich regions. Cortex is B cell rich and paracortex is T cell rich. So paracortex is going to have T cells as well as dendritic cells, because dendritic cells are the ones which are going to do the antigen presentation to those T cells through the MHC molecules. Okay, so we are not going to discuss about the activation of T cells and B cells here. We will be talking about that in detail in the lectures specifically for those B cells and T cells, right? Activation lectures. Right now, what I want to tell you is that which regions, what is the histology of lymph node, and which regions is rich in which type of cells. Okay, so I hope the lymph node schematic, the schematic is clear to you all. So how does antigen enters in this lymph node? It is going to enter through the afferent lymphatics, and if it enters through the blood, it is going to get trapped in the capsule. Okay, so either way, it is entering in the lymph nodes through the lymphatics, and once it enters, it is going to it is going to face the lymphocytes for first time in its life. Okay, tissue resident antigen, or either if either the antigen could enter, okay, simply the bacteria could enter here, or instead of those antigens, the antigens could be presented through the antigen presenting cells. So either way, either antigen is there or the antigen presenting cells, which will have MHC molecules, which are presenting the processed antigenic peptides over them. They will be presenting those to the lymphocytes. So either way, the lymphocytes are going to get activated in the lymph nodes, and once they get activated, they are going to leave using the efferent lymphatics. Okay, so I hope this whole pathway is clear to you all, how the antigen enters and how they will be tackled. Okay, so I want to discuss one more point here about chemokines. Okay, so chemokines, they are basically once the lymphocytes or once the immune cells get activated, they secrete chemokines. And what does chemokines do? The chemokines set a gradient. So there will be chemokine gradient across the tissue, and that is going to help the immune cells to navigate and to know about where the need of the immune cells is. So for you to navigate through the areas, you have eyes. So humans can navigate and they can decide where they want to be. But immune cells, how will they know that in that particular region, that particular tissue is going to need them? So that will be done with the help of chemokines. So chemokines, this grade. So let's say that this is the tissue. Okay. So let's say that this is the tissue, and in this particular tissue, the infection is seen. Okay. So this particular part is infected. So the immune cells which are present in this part, they are going to secrete these chemokines. Okay, so those chemokines molecules will be secreted by these immune cells, and that is why there will be a gradient. There will be seen a gradient of chemokines. So chemokine gradient will be seen. So here will be higher chemokine, and here we will see low concentration of chemokines. That is why this will help the immune cells which are residing over the tissue in the other parts which are not infected yet. They will be, they will get those signaling molecules, chemokine, and they will see that the chemokine concentration is higher in this region, means the immune cell requirement is high in this region, and that is going to alert the immune cells to migrate to that region. So chemokines will be secreted either by the immune cells, it can be secreted by the stromal cells. Okay, so granulocyte, macrophages, or stromal cells, they are going to secrete those small signaling molecules, chemokines, that is going to form the concentration gradient across the tissue, and that will help the immune cells to trail and to guide to the specific locations. Okay, so the receptors for the chemokines, they are present over the immune cells, and chemokines, they are secreted by the stromal cells or the immune cells such as granulocytes or macrophages. Okay, so I hope how the so now you know how the immune cells know the location of the infection. So once the immune cells travel to the site of the infection, they are going to mediate the killing. Okay, so I hope the lymph nodes are clear to you all, how the lymph nodes help into the immune responses. Initiation of immune response is clear to you all. So we have discussed about spleen, and we also discussed about lymph nodes in this lecture. In next lecture, we will discuss about mucosal associated lymphoid tissue. So it is going to be a short lecture about discussion about this tissue and how this tissue helps in the initiation of the immune responses, how it creates an environment so that those lymphocytes can react with the antigen, they can interact with the antigen, and they can initiate either the innate immune responses or the adaptive immune responses. So I'm done. I conclude the lecture here. In next lecture, we'll learn about math. Till then, see you. Bye bye.